So this video is part of my sort of biology revision for A-level, and today we're going to be talking about biological membranes. This is in the AS and it's in year 1 for A-levels, so let's get started. So cell membranes are partially permeable, so that basically means that they only let certain substances to enter and leave the cell. The role of cell membranes at the surface of the cell, so that is the cell surface membrane, it separates the cell's content from the external environment. It acts as a barrier for large polar substances. It can act as antigens, which is important for cell recognition in terms of the immune system. Basically, it's basically when the immune system recognizes the cell membrane or the cell as self and not as a foreign cell. It has receptors for cell signaling, and it may be the site of chemical reactions, and it also may contain enzymes that are involved in metabolic reactions and metabolic pathways. So in terms of the role of membranes, of cell membranes within cells, we are particularly looking at organelles. So organelles such as mitochondria and chloroplasts, they have cell membranes. So that's what we are talking about when we are saying within cells. So points that you could put in if it does come up in an exam is for compartmentalization, which basically separate content in the organelles from the cytoplasm. So lysosomes, for an example, they contain hydrolytic digestive enzymes, and having a cell membrane isolates it and basically prevents the digestive enzymes from disrupting other part of the cell, if that makes sense. So kind of isolates them. As it's also a site of chemical reactions. It provides a surface for attachment of enzymes. It controls which substances enter and leave the organelle, and also for intracellular communication. So the model used to describe the structure of a cell membrane is the fluid mosaic model, and this model is just used to describe the structure of the cell membrane. So the fluid bit basically shows that the components of the cell membrane, the proteins and the phospholipid molecules, are floating within it and can move freely, and the mosaic part of it suggests that the components within the cell membrane make up a sort of scattered pattern, which looks like a mosaic, if that makes sense. So now we are going to be looking at the structure of the cell membrane and talking about the different components that actually make up the cell membrane. So the first thing we're going to talk about are phospholipid molecules. So as you can see from the picture to the right, you can see that the phospholipid molecules form a phospholipid bilayer. So within a phospholipid molecule, you have a phosphate head and you have the fatty acid tails. And the phosphate head is hydrophilic. So hydrophilic basically means that it is attracted to water, so the phosphate head face towards water. Whereas the fatty acid tails, they are hydrophobic, and this means that the fatty acid tails basically repel water and face inwards away from water, and this basically forms a phospholipid bilayer. And the phospholipid bilayer, it acts as a barrier for large polar substances. So channel proteins and carrier proteins, these are intrinsic proteins. So that basically means that these proteins are within the cell membrane. So they span through the cell membrane, as you can see towards the right. Like the picture basically shows that they are within the cell membrane, if that makes sense. So channel proteins assist the movement of polar molecules and ions to pass through the cell surface membrane, as the phospholipid bilayer provides a barrier for polar and large substances. That means that they can't just pass through, they have to go through channel proteins and or carrier proteins. And similar to channel proteins, as I said, carrier proteins also assist the movement of larger molecules to pass through the cell surface membrane, such as glucose. And carrier proteins do this by changing their shape, which I will talk about later on. Some proteins may be attached to carrier proteins and function as antigens for cell recognition, enzymes, or receptors for cell signaling. And there are also extrinsic proteins which are located on either side of the phospholipid. So that means it doesn't like run through it, if that makes sense. So next component we have is cholesterol. So cholesterol controls the fluidity of the membrane by preventing the phospholipids from packing too closely together. Cholesterol basically increases the mechanical strength and stabilizes the membrane. And in terms of its location, as you can see from the picture on the right, you can see that that cholesterol is located between phospholipid molecules. Glycolipids and glycoproteins. So glycolipids are carbohydrate chains that are attached to a lipid, and glycoproteins are carbohydrate chains that are attached to a protein. Glycolipids and glycoproteins can act as receptor molecules for cell signaling. So in terms of the binding, so it acts as the binding site for hormones and neurotransmitters, and cell adhesion. So cell adhesion is the formation of hydrogen bonds with water molecules surrounding the cell. Glycolipids and glycoproteins can also act as antigens for cell recognition. This is so that the immune system can recognize the cell as its own and not attack it. It is important for you to remember that not all cell membranes have the same composition. So in neurons and nerve cells, there is typically a higher composition of channel proteins and carrier proteins along its axons, and that is to allow ions such as sodium and potassium ions to enter and exit the cell. White blood cells, plasma membranes typically have a higher composition of glycoproteins that act as receptors to recognize antigens on foreign cells and pathogens. Root hair cells, plasma membranes have a higher composition of carrier proteins to actively transport nitrate ions into the cell from the soil. So diffusion is the net movement of molecules from an area of high concentration of that molecule to an area of low concentration of that molecule, or you could just say is a net movement of molecules down the concentration gradient. This does not necessarily have to be across a cell membrane. It is a passive process, so that means it does not require energy from ATP, but it does rely on the kinetic energy of the molecule. So within diffusion, you have two different types of diffusion. You have simple diffusion and facilitated diffusion. So simple diffusion, it's basically where like some molecules such as oxygen and carbon dioxide, which are small and non-polar, can literally just pass through the cell membrane by diffusion. So this is simple diffusion. Lipid-soluble molecules such as steroid hormones can also diffuse through the cell membrane by simple diffusion. However, polar and larger molecules cannot pass through the phospholipid bilayer, so transport proteins such as channel proteins assist the diffusion of these molecules across the plasma membrane, and this is facilitated diffusion. It's basically diffusion but with the help of a transport protein. So osmosis is the net movement of water molecules across a partially permeable membrane down the water potential gradient. Instead of partially permeable, you can say semi-permeable. It still means the same thing. Water potential is a measure of the tendency of water molecules to diffuse from one region to another. Pure water has the highest water potential, and the water potential decreases as more solutes are added to the solution. Once the water potential on both sides become equal, there is no net osmosis, but water molecules are still moving around randomly, so they could like swap places, but there'll just be no net osmosis. Water potential is measured in kilopascals, and the highest value for water potential is 0, which is the water potential for pure water. And as more solutes are added to the solution, the value of water potential becomes more negative. So the more negative the value, the lower the water potential. So now we are going to be talking about the water potential of cells. We're going to be talking about hypotonic solution and hypertonic solution. There is a third one, which is isotonic, but that one, in terms of what you need to know, is just basically when the solution, the water potential in the solution and the water potential in the cell are basically the same. So water is kind of like moving inside and out. So nothing too much about it, to be honest, but I just want to talk about hypotonic solution and hypertonic solution in a bit more detail. So let's go. So in animal cells, a hypotonic solution in general, it just means when the water potential of the solution is lower than the water potential inside the cell. And in animal cells, this means that water leaves the cell by osmosis across the partially permeable plasma membrane, and the animal cell actually shrivels and it is said to become crenated. Whereas in plant cells, it still means the same thing in the sense that water still leaves the cell by osmosis across the partially permeable plasma membrane. The cytoplasm of plant cells shrink and the membrane pulls away from the cell wall of the plant cell, and the plant cell is said to be plasmolysed, and the tissue of plasmolysed cells is described as flaccid. In a hypertonic solution, the water potential inside the cell is lower than the water potential outside the cell. So water moves into the cell by osmosis down the water potential gradient across the partially permeable membrane, plasma membrane. In animal cells, because a lot of water molecules are entering the cell will eventually swell and it will eventually burst. And this is called cytolysis in animal cells. Whereas in plant cells, the cellulose cell wall actually prevents the cell from bursting. So when the cell does swell up because a lot of water molecules are entering the cell by osmosis down the water potential gradient, it will push against the cell wall, and that basically prevents the cell from bursting. The plant cell is said to be turgid, and this can actually be beneficial because it provides the plant cell with support. So now we are looking at active transport. So active transport is the movement of substances against the concentration gradient across a cell membrane using ATP and carrier proteins. The energy released from ATP actually allows the carrier proteins to change its shape. So bulk transport. Some cells need to transport large molecules which are too large to pass through the plasma membrane. So bulk transport is used to transport these large molecules. Bulk transport requires energy from ATP. So within bulk transport, there are two processes that you need to know: endocytosis and exocytosis. So Endocytosis is a process that shows how large molecules are transported into the cell. So phagocytosis and pinocytosis are types of endocytosis. ATP is needed to provide energy to form the vesicles and move the vesicles by using motor proteins that move along the cytoskeleton. Exocytosis is a process that shows how large molecules are transported out of the cell, and the vesicle travels towards the plasma membrane and fuses with the plasma membrane, and then its contents within the vesicle is then released out of the cell. So here is a diagram basically showing the process of endocytosis. So as you can see, the sort of cytoplasm, well, the cell membrane kind of like surrounds the large molecules, and then it fuses to form a vesicle, and then the vesicle is transported to where it needs to go via motor proteins, which are on the cytoskeleton. So here is a sort of diagram picture of exocytosis. So the vesicle travels towards the plasma membrane and it fuses with the plasma membrane. And once it fuses with the plasma membrane, that is when the contents within the vesicle is then released out of the cell. So now we are looking at the effect of temperature on the permeability of the membrane. So the first thing we're going to look at is when the temperature drops, so a decrease in temperature. So a decrease in temperature causes the phospholipid molecules to have less kinetic energy, so that means that they are moving more slowly, and that decreases the permeability and fluidity of the membrane. The saturated fatty acids are compressed, whereas the unsaturated fatty acids actually push adjacent next to phospholipid molecules away because of their kinks, and this helps to maintain the fluidity of the membrane. So it basically makes the phospholipids to be not as compressed. And that basically shows that the proportion of saturated and unsaturated fatty acids within the phospholipid bilayer can actually determine the fluidity of the membrane when it's under lower temperatures or colder temperatures. Cholesterol also prevents the phospholipid molecules from packing too closely together, so it also helps to maintain the fluidity of the membrane. An increase in temperature causes the phospholipid molecules to have more kinetic energy, so that means they are moving a lot more. And this increases the fluidity and the permeability of the membrane. However, if the temperature increases too much, then proteins that are embedded within the plasma membrane would denature, and that is because the bonds such as hydrogen bonds and ionic forces that hold the tertiary structure of the protein together are broken. So that means that the protein changes shape and can no longer do its function. The cytoskeleton underneath the plasma membrane also denatures because the cytoskeleton is also a protein. And this all basically means that the plasma membrane basically falls apart and it is damaged. And therefore, the plasma membrane becomes even more permeable because of that. So the next thing we are going to talk about is the effect of solvent on the phospholipid molecules. So organic solvents such as ethanol damages the cell membrane because the phospholipids dissolve in the solvent. So thank you guys for watching this video. I hope you guys found it helpful and useful, and I hope this was a good revision video on biological membranes, and I hope you guys have a good day, a great day, and I will see you in another video. So yeah, bye guys, bye.